Hello, everyone, and welcome to today's webinar in the Public Health webinar series on blood disorders. This webinar is presented by the Division of Blood Disorders at the Centers for Disease Control and Prevention, also known as the CDC. I'm Dr. Steve Pipe, and I serve as Professor of Pediatrics and Pathology at the University of Michigan. I'm the Pediatric Medical Director for the Hemophilia and Coagulation Disorders Program and direct the Special Coagulation Laboratory. I also serve as Chair for the Medical and Scientific Advisory Council for the National Hemophilia Foundation. I'm delighted to be joined by my colleague, Dr. Maggie Ragney. Dr. Ragney is Professor of Medicine and Clinical and Translational Medicine and Vascular Medicine Institute in the Division of Hematology and Oncology at the University of Pittsburgh. And she also serves as Medical Director at the Hemophilia Center of Western Pennsylvania in Pittsburgh. Well, we're both excited to be with you today to share some of our current thinking and next steps on the state of the science on inhibitors. Now, before we begin, I'd like to go over a few points and helpful tips for joining this webinar. So upon joining the webinar, you'll see two audio options. You can opt to listen via the speakers on your computer or by using your phone to call in. And if the phone option is chosen, the webinar toolbar will provide you with the phone and access number and audio pin. All audio will be muted, and at the end of the presentation, there will be a time for questions. You'll be able to submit a written question. You please use the questions area of your toolbar, and we'll respond to as many questions as our time allows. Today's webinar will last approximately 1 hour. Please note that this webinar will be recorded. And we'll begin today's webinar first with two brief audience polls. First, we're interested in knowing more about you. So what's going to appear on the screen is a polling window. And so we want to know what best describes you. So select your best characterization of who you are today. And we'll give everyone a chance to make their selections. And then in just a few more seconds, we'll be able to see the polls. All right, so it looks like we have just under half of our participants today consider themselves healthcare providers. We've got some public health professionals. We've really pleased to see some persons with bleeding disorders participating today, and then we've also got our colleagues from industry, so that's terrific. We're now going to do one more poll, get a little bit more data on everybody. So we want to see what geographies we're covering today. So which region of the world are you from? And they should be wrapping that up for us shortly. And then let's just see where our various geographies are. Well, it looks like the predominance is U.S., Canada, Mexico. But we do have some participants from Europe as well as from either Asia or Africa. So that's terrific. So welcome to everybody this afternoon or morning or evening, depending on where you're at. So this particular webinar is entitled Inhibitor Prevention and Eradication, from the NHLBI State of the Science to the INHIBIT trial. And you'll hear about both of these between myself and Dr. Ragney. So just to set the stage for what the problem is with inhibitors. So in the context of severe hemophilia A for individuals who make no measurable factor VIII protein, upon exposure to any form of factor VIII, it's essentially a normal immunologic response to mount antibodies against that exogenous factor. And this occurs in roughly 30% of individual severe hemophilia A, but depending on the trials, that has ranged even as high as into the 40% range. We know that these involve T-cell dependent B-cell responses. These are allo antibodies to the exogenous factor VIII, and in sufficient titer, they will prevent factor VIII from functioning in hemostasis. This requires the use of bypassing agents, either recombinant factor VIIa or activated prothrombin complex concentrates, or FIBA, for acute bleed management. Now, in the last couple of years, at least in the U.S. and many other countries around the world, we've had access to emicizumab, which works as a prophylactic agent for bleeding. But patients with inhibitors on emicizumab still have to rely on 7A or FIBA for management of breakthrough bleeding. The overall morbidity of inhibitors has been widely described. They have poor control of bleeding. They have almost twice the hospitalizations, 10 times the cost, and 3.5 times the mortality. Emicizumab has made some dent in some of those outcome measures, but also requires a significant annual cost. So the goal that we're going to be talking about today is how can we both prevent and eradicate inhibitors to allow patients to resume regular factor VIII replacement and to treat bleeding if needed. Now, the origins of this workshop are from within the CDC's Division of Blood Disorders. One of their commitments is the goal of reducing the occurrence of inhibitors, because this is the most significant and costly complication affecting people with hemophilia today. And in March 2012, we held the first inhibitor summit. This was a multi-stakeholder engagement, and this informed the integration of the inhibitor surveillance that's now incorporated into what we call the Community Counts Registry for Bleeding Disorders Surveillance. This is funded by the CDC through a cooperative agreement that was awarded to the American Thrombosis and Hemostasis Network, or ATHN, in partnership with the U.S. Hemophilia Treatment Center Network. And this collects information about key aspects of inhibitor development, treatment, and outcomes. The Division of Blood Disorders Reference Laboratory also developed the methodology for sensitive and specific inhibitor testing, which is performed on all eligible registry participants, and also offers testing for those who are on emicizumab. In January of 2017, we had the second inhibitor summit. Now, at this summit, we recognized the need for collaboration across the entire U.S. bleeding disorders community, including key government, scientific, clinical, and other bleeding disorder community stakeholders. And the objectives of this second summit were to share information about the registry and the current state of our national inhibitor surveillance, to identify steps to maximize the accuracy and representativeness of national data on inhibitor occurrence, and to determine the strategy to maintain the accuracy and validity of inhibitor testing methods. But one important objective that came out of this second inhibitor summit was the need to explore a national coordinated inhibitor science agenda. So this charge was driven by these important points as it relates to understanding inhibitors. The subjects that are needed for studies in this area are primarily previously untreated patients, and essentially they are infants and toddlers. This is a very precious resource, and so efforts really should be made to coordinate the studies so that we can maximize the benefit that can be obtained from each study in the subject. It's also important to ensure that really only the best science is performed, and that funding for studies should be adequate to cover the costs of obtaining really high quality data. It was felt that multifaceted education and marketing activities would need to be directed to the patient community well in advance of upcoming trials in order to stimulate interest and participation. And the need to develop a multidisciplinary group that was working to develop and implement an integrated scientific and public health agenda, as well as to establish definitions and common data elements. It was thought that representatives from a wide variety of disciplines should be included in order to facilitate the generation of new ideas and approaches, and then agreement from the bleeding disorder community to proceed with a regimented series of cooperative studies that had been appropriately vetted. So this charge was given to an inhibitor working group from the MASAC, the Medical and Scientific Advisory Council, to the NHF. And the idea was put forth by the Deputy Director, Donna D. McKelly, from the Division of Blood Disorders at NHLBI, to basically hold a workshop where we would solicit hemophilia community-wide input into the development of a coordinated U.S.-based blueprint for future basic, translational, and clinical research focused on factor VIII immunogenicity and factor VIII inhibitor prevention and eradication. This workshop ended up having 200 registered participants from 29 states and 9 countries. Half of the registrants were from academia and hemophilia treatment centers, 20% were from industry, 18% from the federal government, and 8% represented patient advocacy. Any participants who could not attend in person were able to participate in the workshop through video casting. An executive steering committee was established that had great input from some clinicians and scientists, the NHLBI Division of Blood Disorder Director and Deputy Director, representatives from the CDC, as well as a community and societies representative. And the mandate was to establish the focus and the leadership for the scientific working groups that were going to be set up for this workshop, to oversee the scientific working group activities, and then to develop the State of the Science workshop agenda. So these working groups were assembled. There ended up being 4 working groups, which I'll walk you through, and they met for months deliberating on their charges. And then we met over a 2-day meeting at the NIH in Bethesda, Maryland, in May 15th and 16th of 2018. And we called this the Factor VIII Inhibitors Generating a National Blueprint for Future Research. And this link that is at the bottom there is actually still live. You can go there. There are archived videos of many of the NHLBI workshops, but you'll be able to search for this particular one, and if you so choose, you can actually review the activities that went on over those 2 days. Now, I'm not going to talk about Working Group 1 just yet, because that's going to be part of the lead-in to Dr. Ragney's presentation, because that working group was focused on innovative clinical trials. Working Group 2 was charged with trying to develop 21st century data and biospecimen collection to assist with longitudinal lifespan cohort studies. And this was their observations from their deliberations that there really needed to be more infrastructure support. They identified that we needed to leverage current national data collection efforts. For example, mentioned the community counts and other efforts through AFIN to establish and optimize automated clinical and patient-entered data transfer. Use standard outcome measures across studies that would allow data integration, including from international sources, streamline the protocols, the contracting, the informed consent processes, including the use of single IRBs, and centralized tracking of biorepository specimens and data for timely utilization. There was going to be need for resources and partnerships through the hemophilia treatment centers, as they would primarily be doing the data entry and biospecimen collection. And then there was opportunities to optimize private-public collaborations and funding opportunities. Patient engagement was going to be key. Need to engage patients to identify patient-important outcomes in all stages of research, and also to build strong collaborative relationships with the patient organizations, because they will help inform research questions and also be part of the dissemination of the new knowledge. And then all of this work was also opportunity for training initiatives, which is a key priority of NHLBI. Working Group 3 was focused on basic and translational research studies for understanding factor VIII immunogenicity. And they identified 3 scientific priorities. The first one being more learnings around the activation signals and immune regulation that shape the response to factor VIII. Particular focus on the innate and early immune recognition of factor VIII, more characterization of the adaptive immune responses, and then also immune regulation and tolerance. It was felt that there needed to be exploration of the utility of non-animal models to help predict inhibitor formation, since the animal models often have not properly informed what's happened in humans. And then some interesting studies on how the site of factor VIII expression, its structure, and interactions with von Willebrand factor determine immunogenicity and tolerance. And this includes factor VIII when it's expressed in the context of gene therapy, where it's being expressed within an endogenous tissue like the liver, like we're doing currently with hemophilia gene therapy. What happens when there are native interactions with von Willebrand factor, or what happens if we bypass those native interactions with bioengineered molecules that have altered structure. So the picture here is that a previously untreated patient will at some point start to get exposed to factor VIII. If they have a series of exposures with no development inhibitor, we would call them a previously treated patient. And those patients may ultimately go on not just continue on exogenous factor VIII exposure, but may actually be candidates for gene therapy. If a pup develops an inhibitor, they typically have been offered immune tolerance induction, and then there's sort of two outcomes there. They could be successful, in which case they could resume factor VIII prophylaxis, or they could fail. And there is some question about whether these refractory inhibitor patients could also be managed with gene therapy in the future. That hasn't yet been launched, but that has been proposed. And then related to the factor VIII immunogenicity, looking at this from a number of different scenarios, the genetics, single cell analysis, in vitro experiments, analysis from patient blood samples, animal models, and so-called in silico or computer analysis to inform about factor VIII immunogenicity. The final working group was actually quite interesting, and they were charged with the design of an antenatal neonatal cohort for translational research studies. And their deliberations and interactions with the scientists in their group had identified that it's possible that the path for the development of inhibitors or the development of tolerance may actually be influenced by neonates while they are still in the womb. And so the principle here that's being laid out in this graphic is that mothers could be identified and consented. Prenatal testing would determine if they were carrying a male fetus who had a factor VIII gene variant that was known to be associated with inhibitors. That at-risk pregnancy could then be enrolled during the antenatal period, where maternal blood sampling could be performed with different biomarkers, and immunologic analytes could be analyzed. Upon delivery of the neonate, samples from both maternal blood, placenta, and neonate could be performed, and then that neonate could now be followed in a longitudinal cohort study with longitudinal blood sampling, trying to identify the impact from that first exogenous factor VIII exposure during infancy, and then with repeated exposures over early childhood and later in life. And then once that once an inhibitor had formed or tolerance had been demonstrated, that infant may then be eligible for other studies. This is the overall schema that came from all the working groups working together. So we've already talked about the idea of a new way of thinking about understanding immunologic development in response to factor VIII may be what's going on in the mother and for the baby in the womb. So we need to identify known carriers, but there could also be de novo mutations. Once a new diagnosis of hemophilia is made at birth, that baby is going to start in what we're going to call these swim lanes. So the baby's going to start in the yellow swim lane, which is that baby's going to be at risk for inhibitor, and we don't know what's going to happen. That baby could be part of an observational cohort where we could be looking for positive biomarkers that might predict inhibitor development. And then with exposure to factor VIII, there's going to be one of two paths. They would either develop an inhibitor or they would show that they are tolerant. that tolerant infant is equally valuable for looking at the immunologic responses to understand why they were tolerant. And indeed, the others who have developed an inhibitor would have an option of participating in a clinical trial intervention for either inhibitor prevention or eradication if they've actually developed an inhibitor. Once they finish those clinical trials, the idea is that regardless of what their outcome from that trial is, they would return to these cohorts for further studies. The outcomes from these studies would then inform the next wave of infants who come into these cohorts, and that we would have an iterative process to give us insights on what's going on with inhibitor formation. So the scientific priorities here are longitudinal outcomes and surveillance, observational cohort discovery, subject discovery from the clinical trials, depending on their outcome. And then there's this concept of developmental origins of health and disease. This is an interesting concept where exposures very early in life can have an influence on the later development of disease many decades later. So we sort of have this picture of infants all the way through to adults participating in these cohort studies. Now we finally get to working group 1, and this is going to be the balance of our presentation today. And this working group was really given 4 scientific priorities to look at. It include clinical trialists, those who are experts in design, industry partners, and then those who were part of clinician and patient engagement and community resources. And so I'm going to now turn it over to Dr. Ragney, and she can tell you about some of the exciting output from this working group number 1.
Okay, thank you for that, Dr. Pipe. I'm hoping my slides will be coming up shortly. So I'm just going to go ahead, assuming that they have come up. So the general goals for this working group 1 that had the advantage of these experts in trials, design, industry, and from the community were 1, to define what resources and partnerships would be required to facilitate clinical trials. Number 2, to figure out how to leverage and support this HTC infrastructure for clinical trials, and to be sure to embed mechanistic studies into the clinical trials that will help us understand the natural history and correlates of inhibitor formation and tolerance. We also were looking at how to optimize public-private partnerships that would be crucial to conduct clinical trials and to engage the patient community. Without their engagement, clinical trials will not happen. And finally, very importantly, to embed training opportunities within the clinical trials for young investigators to assure that this whole concept can continue on in a very positive way. So on the next slide, I guess I'm doing that. Okay, hold on. So, good. So the specific goals for clinical trials were, number 1, to consider the challenges of rare disease research in this rare population, and then to consider what are the statistical considerations in this setting. For example, we will be talking a little bit about a master protocol concept. And then the concept of how do we coordinate these clinical trials within a trials network to make this an efficient proposition. And finally, how do we design prevention and eradication trials? So let's consider each of these. Number 1, the challenges of rare disease clinical trials. So clearly, patients are limited. In a survey we did in 2017, we calculated there are about 1.82 pups per HTC per year who are born. This is a very small number, but when you consider that the outcomes are even rarer, so we're saying approximately 30% develop inhibitors, we're talking about a very, very rare group of patients that we're targeting for this very critical question, these very critical questions. And on top of that, the competition for these patients is fierce. Pharma, non-randomized trials, and all sorts of other things are competing for patients. in order to do these trials, partnerships are critical with pharma to help us facilitate new drugs that we might want to use in these trials that may be very expensive, government and community support. This is really a partnership to make this work. And what we know is that in rare diseases, classical randomized controlled trials are really just not possible. There are too few patients, too many sites, and the time over which it would take to conduct these trials is extraordinarily expensive. And HTCs themselves require resources. We did a 2018 survey of HTCs. What would it take for you to engage in a clinical trial to prevent or eradicate inhibitors? And we found that 30 to 40% said there was no question that they needed nurse to do the research and technicians to obtain the samples and correctly process them and ship them to the labs that would be doing the very careful, quality-controlled laboratory tests that are needed. And finally, a randomized controlled trial requires a major infrastructure. You need at least 30 to 40 hemophilia treatment centers within an infrastructure to conduct the trials and the partnerships with industry, community, and government to actually conduct it. And finally, we really don't know what the optimal approach to inhibitor prevention and eradication is. The second concept was about statistical considerations. So it's not easy to do these trials in a rare disease population. In fact, you can't do standard randomized trials. So how can you get the statistical considerations to be implemented within your concept so that you can actually run the trial? A couple of the concepts were to use historical data to reduce the sample size. So you could actually use known data on inhibitor prevention or eradication as part of your data in the trial to reduce the number of patients in the trial and even to prioritize randomization to a novel treatment or a treatment that is favored by the community. You could utilize CDC surveillance registry and international registries to follow the natural history and from which to have patients have the opportunity to join the trial or just to continue to be followed to help inform our understanding of how frequently inhibitors are found and eradicated. And finally, to use databases and central labs as a platform to also launch these clinical trials, so that we really are mining the data we have. We're using the patients we have in the most efficient manner. And finally, the concept of a master protocol where you could actually follow anyone enrolled and screened for inhibitor development. And you could establish baseline pre-inhibitor laboratory data, collect specimens, to establish the natural history study among those screened, and from some of them actually then enroll in a trial. So on the next slide, we have the concept of the master protocol. And what you see here is you take all the hemophilia A inhibitor patients born in the U.S. They're all screened for inhibitors, some of whom may enroll in the inhibitor prevention trial, but some of whom may actually just elect to be followed. They don't want to be in a trial or for some reason can't be in a trial. But in all of these patients, whether enrolled or just followed, you can determine inhibitor development, and those who develop inhibitors would have the potential to enroll on an inhibitor eradication trial. So once you've developed an inhibitor, you can enroll on that eradication trial. The concept here is that you are making the best effort and the best chance to utilize these rare patients and to maximize the data that you do find. The fourth issue that we talked about was the clinical trials network. We really don't have a unifying trials network to perform hemophilia treatment, to perform hemophilia inhibitor and eradication trials. This would provide HTCs with nurse coordinators that they've already told us 40% need, as well as the technician support that 40% of the centers need. It would implement a system for standardized biospecimen and data collection, incorporate standardized outcome measures so that across the board we're doing the same thing in clinical trials that are developed. You would incorporate mechanistic studies with quality-controlled labs to study factor VIII tolerance. You would engage the patient community to determine, are these the trials we want to do? Will you participate in these trials? Do these make sense? Are you engaged? Without the engagement of the patient community, we cannot go forward. And in addition, train early-stage investigators to be part of this entire effort, to sit in on conference calls, to be part of the enrollment, to be part of the community engagement, and to help standardize outcome measures and to work with this biospecimen and data collection. And finally, to establish resources and partnerships that will facilitate not only the first clinical trials in prevention and eradication, but those to come with new drugs as they're developed. And we sort of envisioned the clinical trials infrastructure in the following slide here, as you see. So the center of this is the Hemophilia Clinical Trials Group, HCTG, which we really borrowed heavily from the whole concept of what was done in the AIDS Clinical Trials Group, which we had a chance to participate in with the hemophilia community. And as you can see, there is a partnership and input from the government and NHLBI, from HRSA 340B, from consumers, from charities, from insurers, from industry, from community groups and foundations. And we in this working group one actually contacted every one of these groups to talk about what would be the way that could happen. What was envisioned? What would it take to leverage the community to be part of this, the government, the industry, and insurers? So these are critical issues that are required for clinical trials design and implementation. And the fifth issue was to design trials to prevent and eradicate inhibitors, to incorporate novel therapies into trials as they are FDA approved and developed, to review inhibitor prevention and eradication data with each new information that comes along, and then to define the state of equipoise at each time you're ready to start a new trial. Where do things stand? What do we really know about inhibitor prevention and eradication by the time we've gone through a few iterations of clinical trials? And then finally, to incorporate adaptive design into the clinical trials platform. And what we mean by that is use some of the very amazing tricks that trialists use, such as Bayesian priors, which is really incorporating historical data into your trial so that you don't have to enroll 99 or 66 patients, but actually utilize maybe a quarter or a third of the data from existing information. So this brings me to the next slide, which is right now our two most novel therapies are Eloctate and emicizumab. Eloctate, the Fc fusion protein that we know extends half-life. It's given IV. It promotes Tregs, which are T regulatory lymphocytes, and thereby has the potential to promote factor VIII tolerance. Emicizumab is the bispecific monoclonal antibody that mimics factor VIII. It binds 9A and 10, and it's given sub-Q and is highly preferred by patients both with and without inhibitors. And it promotes hemostasis by avoiding factor VIII. So if one were to use these drugs in a clinical trial, what do we know before as we're trying to set this up? So if we're considering inhibitor prevention in PUPs or previously untreated patients, what do we know about these drugs? Well, we know that Eloctate has the potential to reduce inhibitor incidence, but the only data that we have is in a very small trial, the ALONG trial. in which anti-A was detected in about 30% of these pups. That was not the complete data. These are based on data from 2018. There's been no update to my knowledge, but it's a little concerning that it does not appear to reduce it. But we don't have any randomized trials, and we have a very small number of patients. In addition, we have very minimal data that are just case reports, which is probably the worst kind of data you can use, but we're talking about not much data. And in a study that we did in two cousins who had the same mutation, were from sisters who were their mothers, one child who before he ever had a bleed or ever got any factor was given Advate and developed an inhibitor to 43 Bethesda units, whereas his cousin, before he had a bleed or got any factor, was given a loctate. He still developed an inhibitor, but his titer was so much lower, 1.4 Bethesda units. And in fact, in the A-long trial, about half of the inhibitors that developed were low titer inhibitors. So that's a very interesting piece of information. It doesn't give us all the data we want, but that's what we know to date. And what do we know about inhibitor eradication with these drugs? Well, eloctate appears to reduce the time to tolerance, that is to eradication, in some cases to 3 to 8 months. It was about 2.7 months in a couple patients that Dr. Malik and I studied, and it was about 6.9 months in a study that Dr. Carcao studied. These were just median times to eradication. So not all the patients in his study actually had a shortened time, but there were a group who did. We don't really know how they differed from those who did not. Now, what do we know about emicizumab? Well, we know in a study that Batsuli did that some of the patients with previously existing inhibitors were able to have a reduction in bleeds with emicizumab when it was combined with their R factor 8, recombinant factor 8 ITI. But they did not, and I sent another email to find out, and I have confirmed, they did not measure the time to tolerance because the study started within the ongoing tolerance in those patients. And in one patient, emicizumab plus eloctate also reduced the bleeds in tolerized patients, but the time to tolerance, again, was unknown. So we have a lot of unknown information. So where does that put us? Well, we're really in equipoise. What we know is that while eloctate can induce Tregs and promote tolerance, inhibitors still develop in up to 30% based on small amounts of data. And eloctate can shorten immune tolerance to less than 10 months in selected patients, and we don't know why. So that puts us with the question, does eloctate sufficiently reduce inhibitors and shorten ITI to justify its use? So what about emicizumab? While emicizumab provides hemostasis and appears to avoid factor VIII exposure, and in some patients reduces the ABR, the annualized bleed rate, if breakthrough bleeds occur that require factor VIII while you're performing immune tolerance, it could be just as bad and trigger danger and inhibitor formation and slow ITI. So the question is, does emicizumab really reduce or just delay inhibitor formation or shorten ITI sufficiently to justify its use? So we came up with two trial concepts. These have actually been modified, and we're working with these at this point. The first was the inhibitor prevention trial, and the second that I'll show you in a minute is the inhibitor eradication trial. So for the inhibitor prevention trial, our hypothesis was that emicizumab can reduce bleeds and danger, but it's less immunogenic than factor VIII. So our intervention was to compare weekly eloctate versus emicizumab before the first bleed. Primary endpoint would be inhibitor development, measuring anti-8 with the chromogenic NBU and greater than 5 at 48 weeks would determine and define inhibitor development. Of course, many secondary endpoints, bleeding, mechanistic studies, ELISpot, microbiome, and precision medicine now in validated small amounts, working with Working Group 3 and Working Group 2 are critical issues. In addition, we would want to engage mothers, and working with Working Group 4, at the same time this trial would go on, we would be collecting samples from mothers so that we could follow them in different swim lanes, as Steve pointed out, so that we could find out, are there any factors in the pre-birth time that would show us other interesting correlates and markers of inhibitor development. So now on this slide, we're looking at clinical trial number 2, and this is the inhibitor eradication trial. And as you can see here, the hypothesis is that emicizumab reduces bleeds and danger, but it's yet less immunogenic than standard factor VIII. So here our intervention would be eloctate ITI with or without emicizumab weekly in previously treated patients. Inhibitor development here, eradication, would be our primary endpoint. We'd want to see how many eradicated the inhibitors they had by 48 weeks, and we would use the very same secondary endpoints, bleeding, mechanistic studies, ELISpot, microbiome, precision medicine, as in the first trial. But one of the things that we realized was the sample size that we would need to do these was huge. So we worked with a group through an XO1 NIH grant, the Berry Consultants, experts in clinical trials in rare disease. And what we developed was an inhibitor clinical trials platform. This would allow us to use what we call Bayesian priors, that is, historical data, to offset the total number of patients enrolled in the trial and to preferentially 1-to-4 randomize patients to emicizumab over eloctate, because patients like those drugs and they're easier to use. So you would start out in the INHIBIT prevention trial, that's the top line, with pups. Randomize them for 48 weeks to eloctate or emicizumab and look at the time to inhibitor development. In those who develop inhibitors, they could then immediately, in the same sites and in the same way, be enrolled in the eradication trial. We designed the two trials to have the same endpoints, the same data collection times, the same validated small volumes for inhibitor measure and all the other secondary measures. And here we would compare eloctate ITI with emicizumab versus eloctate ITI alone, looking at the time to eradication of the inhibitor defined as less than 0.6 Bethesda units. These trials would be linked, so every site would have the potential, once these both trials are up and ready to go, to enroll in both trials and to use data that we know from historic data to reduce the size. So again, on this slide, we're just going over what are the advantages of an adaptive design. It allows efficient use of historic data. We did simulations, thousands of simulations of incorporating historic data into this design, and found that we could preferentially enroll 1-to-4 randomizing to whatever that novel drug was, and here that's emicizumab. The two linked trials have the same outcomes, the same visits, the same labs, making it simpler for each HTC to do these trials. All would be validated samples for small volumes, and they would use quality controlled laboratories to conduct all of the laboratory tests that would all be exactly the same. We worked with Working Group 2 and Working Group 3 to make sure that would happen. Mechanistic assays to study tolerance. We have also worked with Working Group 3 and 2 on these, and then future incorporation of novel drugs as they became available so that these could continue on once answers were found. So where are we in this clinical trials platform work to get these trials up and going? Well, we have certainly applied for funding. We anticipate hearing quite soon. We have set up a coordinated network with about 31 sites that have been willing to enroll in this trial. It doesn't mean that more couldn't enroll, and I believe that what we really need is some support just for the network, not in addition to the trials, and we're working on that at this moment. We've set up a precision medicine registry within the trial so that proteomics and microbiomics could be easily collected. We need to next engage the community. We are looking forward to holding town meetings as part of the next National Hemophilia Foundation, and we are certainly working within our own regional HTC, who is very much engaged in this whole process and engaging chapters within our region. But there's no reason this has to be exclusive. It needs to be done in all of the regional HTCs that would like to participate. We also will be providing training opportunities for early stage investigators. I'm working with an adult hematologist and a pediatric hematologist right here at the University of Pittsburgh who will run these two trials with me. And set up single IRBs through the University of Pittsburgh with relying agreements. So once you go through this, it's a cinch to move forward with that clinical trial. It's a lot of upfront work, but it's well worth it. And we've built in the cost to do this, and our Pitt IRB is now experienced in doing this, and that would be what we would hope to accomplish. So I really want to thank you for your attention. We will now shift to the question and answer section of the webinar. And again, to submit a written question, please use the questions area in your toolbar, and we will certainly be willing to try to answer as many of Dr. Pipe and myself will be very happy to answer any questions with the time remaining.
Maggie, maybe I could pose the first question to you. So could you speak a little bit more about how INHIBIT could be integrated with some of the other PUP observational cohort studies that have been initiated through ATHEN?
Well, I think that would require engagement of ATHEN into this process. In other words, a collaboration. So if patients have been enrolled in that ATHEN, for example, ATHEN working group, or ATHEN number 8, I believe it is, it would be something like this, I imagine, and I'd be very interested in working with the ATHEN 8 group or any others. But I would think that at the time you're ready to enroll someone for long-term follow-up, which is what happens in ATHEN 8, you might pose the question to the family, we want you to be followed up, but if you have an interest in participating in a clinical trial, you could both be followed up and participate in a clinical trial. Just as we showed that master protocol, I think what we really need to have that work is intense collaboration. That is, that any site that wants to enroll in ATHEN 8 might either decide that it also wants to do the clinical trial or would like to refer those patients to the clinical trial itself. We will have 31 HTCs, we hope, open and ready to enroll, but it should not be exclusivity here. We would open it up to as many as would be interested in doing this trial. The cost comes in with the more IRB approvals and all that business, but quite frankly, we're going to work with another group to set up the trial network, and I think through that, by its facilitating the cost of the nurse, 40% cost of a research nurse, and laboratory data collection, a technician to do that, I think it may facilitate it. I would entertain, once we get funding, to talk to Athens 8, to talk to any of the Athens leadership. And I think Mike Recht is very well aware of what we are doing as a site. He previously at Oregon, is well aware of this. But I think it takes collaboration. I think it takes a little bit of effort. But I think what we need to do is have these community discussions, whether it's with Athens or the patients or the insurers or the pharma. I think we need those discussions to make this happen. And I'd be interested in what others think as well.
Yeah, I think that still fits with the schema we talked about from the overall state of the science. This idea is that observational cohorts can be quite broad and could and maybe should be all-inclusive. But from that, individual patients can participate in clinical trials, and then when they're finished with that clinical trial, they return to those observational cohorts. And then the results of so I think there is opportunity for integration here. There was one quick question had to relate to, you're proposing using ME for prophylaxis in the inhibitor patients along with a loctate ITI. How would you manage breakthrough bleeding in these patients given the potential complication?
Right. Well, that's a great question. And the way that we wrote this into the trial was that 7A would be, in an inhibitor patient, the best approach in this sense. It's not an easy drug to use. However, a couple things might be in its favor. I'm worried that if we would use FIBA, we would have potentially more adverse events. In my clinic, we have converted almost entirely to 7A. I realize not all sites may have done that, but for the purposes of the trial and safety issues, we've set it up so they would use 7A. Once the inhibitor is eradicated, certainly they could return to using a loctate or whatever factor VIII drug they wanted to use. But we discussed this in our group, and we thought that would be the safest approach.
The other question that was just submitted was, as you know, there is another factor VIII prevention study that uses a specific product, Nuvic, along with emicizumab, to look at the development of inhibitors over time. So a slightly different format, but in principle maybe similar. I see in your trial that you have the additional biology of the Fc fusion and its potential immunomodulatory perspective. What's your thoughts about, is there room in the U.S. HTC networks for two trials sufficiently powered to answer the questions that these two studies are proposing?
Right. Well, I think we should do as much as we can. I would argue that a randomized trial design will give you much more information than a single arm approach. And so I would actually be biased towards randomized trial design. In fact, NIH supports that approach, and that's the approach we took. I would think that the faster we could get a randomized trial enrolled, the faster we could add new agents to the platform and continue the trials forward. I think that is really once we have the network going and the platform set up and a steering group to discuss next steps, I think that might be the most efficient way. I would never discourage anyone from looking at different drugs, but it may be that given our very small patient population, we must be efficient and we must answer trials to the best of our ability, because if we run a trial and haven't done a comparison, we will really never know what the answer is, and it's difficult then to go on. By the way, this was the way that the AIDS clinical trials groups worked. We had consumers in every working group who demanded randomized trials, and they demanded rapidity, that is, getting people on the trial and moving forward, because they had their lives at stake. And they were very efficient at doing that. And so I would challenge our community and our hemophilia centers to get that kind of activism on behalf of solving this very dreadful problem called inhibitors.
There's another quick question about any specific plans to incorporate gene therapies in development into studies of inhibitor eradication. There is indeed, and there's been at least one protocol proposed, although the details are not available about what sort of vector system it would be used. But I think people have been thinking about this. There's clearly animal studies to show that tolerance can be induced, at least from dog studies, and there's some good immunologic support that endogenous expression probably is tolerizing. So I think after we get through this first wave of gene therapy studies, almost certainly there will be some trials that are going to be allowing patients with active inhibitors or refractory inhibitors.
You know, that's absolutely incredibly positive, and I think what would be wonderful is that we would be far enough along in this trial that we would be able to say, let's add that, let's use that now as our comparator arm. I think those sorts of studies would be exciting. I know Lindsay, I'm blocking on her last name, she'll kill me, but anyway, at Penn, really was on our working group and had some incredible ideas about how best to do this. And I don't know whether it should be within a pharma based trial or whether at some point it would be open to being in a platform such as what we've suggested.
Yeah. There was someone has pointed out here that ATHEN-8 as it's structured, which is the ATHEN pup trial, only follows pups until the development of an inhibitor, and that is correct. What I think we've taken from the state of the science is stopping the scientific evaluation at the point of inhibitor formation. We're missing a lot of important information. So the patients who fail to develop an inhibitor and those that develop an inhibitor and then what their intervention is and their ultimate outcome are all really valuable scientific efforts. And so we really want to have a much more comprehensive follow-up system that truly follows individuals from infancy all the way through into older age.
Well taken point, and I would actually argue that a master protocol would be perfect for that ATHEN-8 if that were a possibility of adapting the enroll and either enroll in a trial or just maintain follow-up and continue past the inhibitor. Absolutely.
Just a point of clarification from another question. Yes, the studies we've talked about today, and really the focus for the entire state of the science really was on factor VIII biology and factor VIII immunogenicity as it relates to hemophilia A. This is the vast majority of all of our inhibitor population. We do recognize that inhibitors can happen in a small percentage of patients with hemophilia B, as well as other bleeding disorders, including severe von Willebrand disease. But those are going to take unique. Those will have to take unique approaches. A similar design to what you laid out probably is not going to be ideal for an inhibitor patient. Now, to that end, another question that came through is with some of the future therapeutics that are coming out, these are these so-called cross-segment agents like fitusiran, which knocks down antithrombin. There have been monoclonal antibodies that target tissue factor pathway inhibitor, and there's a new serine protease inhibitor that targets activated protein C, just started clinical studies. All of these cross-segment therapies at least give the option that the patient's hemostatic efficacy may be no different whether they have an inhibitor or not. And I think while that is true, one thing we've learned with the first cross-segment therapy, if you like, emicizumab, is that there is a difference of risk, however, depending on whether an inhibitor is present or not. The thrombotic complications and microangiopathy that was seen in the early trials with emicizumab seemed to be associated with use of higher doses of FIBA, and the reason those patients were receiving that were because of an inhibitor. In the non-inhibitor trials, there were not any of those same thrombotic events and TMA events. So I think this is probably also going to be seen with the other cross-segment platforms is that it's possible that although the hemostatic efficacy of the agent and its mechanism of action may be unaffected by the presence of inhibitor, the risk for the patient may be different. And so I think there's still rationale here to identify mechanisms by which we can truly either prevent or eradicate inhibitors over the long haul. Well, I think we're going to wrap up. So, Maggie, I think you have some closing remarks.
I do. So this webinar was brought to you by the CDC's Division of Blood Disorders. We thank the Hemophilia Federation of America for hosting today's webinar. If you have any questions about this series, Cynthia Sayers can be reached at cdc.gov. And this webinar will be archived at the following HTML. And then finally, we would like to bring to your attention that in a few short weeks on March 5th from 2 to 3 p.m., Holger Schoenemann and Andrea Darzi will be talking about selection of a risk assessment model for VTE prevention in hospitalized medical patients, a hot topic. So thank you for your participation today.